This is lecture 2 from the chapter 15 microbes. We're still talking about bacteria. If you haven't watched the Amoeba Sisters bacteria video that is linked on Panther Learn, I would recommend that you go ahead and watch that because I'm going to be referencing that video here and also skipping over some things or going through things pretty quickly because they've already talked about that in their video. So here we're starting with talking about bacteria and their genetic information. So in the video they talked about that bacteria have those chromosomes, the circular chromosomes, and that they also have plasmids, and that these plasmids can kind of be like superpowers. So what kind of powers can the plasmids give a bacteria? So one of the options is a plasmid can be in the metabolic group, meaning that it carries genes that can allow the bacteria to break down new materials. So what does that mean? It means that they now can use new food sources. So let's say before the bacteria could only use sucrose as its energy source. Now maybe that bacteria can use starch as an energy source or fructose, something that it couldn't use before, which will allow it to be able to grow and reproduce faster. Another option is that it could be what we call a resistance plasmid. So resistant plasmids carry genes that allow that bacteria to resist antibiotics in some way. So there are lots of ways for bacteria to be able to resist antibiotics, and so the combinations of genes will allow it to do this in some different way. The last one is what we call virulence plasmids, and this is what controls how sick it can make an organism. So the thing is with bacteria, they want to be able to survive if they're a bacteria that relies on other organisms for their food or a place to live or so forth. Virulence plasmids can be a problem or they can be a benefit. If a virulence plasmid makes a host too sick, that the host actually dies before that bacteria can be moved from one host to another, that's going to be a problem. The kind of key here with a virulence plasmid is to make your host just sick enough that you get passed on. So that all kind of depends on the combination of genes that are in that virulence plasmid. So how do these plasmids get passed? Well, the video showed you two methods. One is the one that they talked about with conjugation, where one bacteria makes a copy and transfers it over. Now, the one thing I want to point out is this shows you too that plasmid being copied and transferred over, but as it says here, it doesn't have to be the plasmid. It can actually be part of the main chromosome that can be copied and transferred over as well. So conjugation is transfer between one bacteria and the other. This is one that the video didn't talk about, so I want to briefly show you this. This is the process of transduction. So transduction is where a virus is actually going to infect a bacteria. And so the way that this works is that the virus's DNA is going to get incorporated into the cell. So this DNA that the virus is infecting the cell with is going to actually be put into the chromosome. And sometimes those genes actually get left in the bacteria if the bacteria doesn't get destroyed by the viral infection. And so that can actually be helpful to the bacteria. In fact, we know this works because not only in bacteria, but even in our own cells, we have the remnants of viral DNA. And some of that viral DNA is being used by organisms today. So this is a very effective way of getting genetic information. And then the last one that they talked about is the transformation. That is the bacteria just picking up pieces of DNA from the environment. Very often this is because of other bacteria being destroyed. So it's not just like random DNA just floating around. Well, it kind of is, but that's why it's there. And so they may be able to pick up genes that they didn't already have and be able to incorporate that in. So this is how bacteria get around that problem of using something like binary fission and making identical copies of themselves, because they can use these three methods of picking up new genetic material to be able to undergo natural selection and evolution. So I mentioned that bacteria are probably the most diverse organisms, and one, it has to do with that genetic exchange. This is another reason, and that is they are the most diverse as far as their metabolism, meaning what they can eat. So I'm not going to ask you to know these names like chemoorganotroph and chemolithotroph and photoautotroph, but I would like you to be familiar with what kinds of things bacteria can eat. So this group over on this side, these guys feed on organic molecules, anything that was once alive. So what you're looking at in this picture, if you can't kind of figure out what you're seeing, this is a shower. And so you've probably seen this stuff before, this kind of an orangish color, maybe sometimes it's a pinkish color in your shower, film that grows in your shower. These are bacteria who are feeding on your skin cells, your soap scum, your soap residue, things like that that are left over. This would also be any of the decomposers, the ones that are eating dead organisms or feces, anything that's left over like that. Chemolithotrophs, these guys, what you're seeing here, this is an old ship, an old shipwreck. This is all metal. And so chemolithotrophs, these ones are actually eating inorganic molecules, so things that weren't alive. They tend to eat things like metals or sulfur or hydrogen molecules as their fuel. So like you see here, these are the rusticles on a ship. You've probably most famously seen these on the Titanic. So they're breaking down iron here. And you can also actually see chemolithotrophs, these inorganic molecule eaters, in mines. So they actually make some spectacular colors in acid mine drainage. And we also see these at hydrothermal vents, those underwater volcanoes, consuming sulfur compounds and hydrogen compounds. The last group are these guys over here. They're green, and they're green for a reason. That's because these are doing photosynthesis. These are our bacteria who can do photosynthesis, which I actually have a little stuffed version of. Isn't it cute? So this is one of the cyanobacteria, the ones that can do photosynthesis. It's actually probably in that little pond scum right there. There's actually quite a few of them that can do it, and these are actually the first organisms on the planet that could do photosynthesis. So we're actually very thankful for them for actually showing up, because they were the first ones to make oxygen on the planet. Now, the video also mentioned that bacteria can be good or bad, harmful or beneficial. So I want to just briefly look at that a little bit and also show you some examples of different diseases. They showed some of them in the video. So you might be familiar with the fact that bacteria are beneficial. So those of them that live in yogurt have been shown to at least take up temporary residence in your digestive tract. So that can be helpful to you. We actually have a large amount of bacteria that live in our digestive tract, a couple pounds of them actually, helping us take out nutrients out of our food, but also they provide us with vitamins that we can't actually make ourselves. And so without them, we would actually have some problems. So they actually are helping us as far as not just getting our food broken down, but actually making some important chemicals that we need. In fact, they don't just live in our guts, but as the video mentioned, they also take up space on our skin. These are what we call our normal flora. So the normal flora, one, live in things like our intestines and help us break down food. But two, they take up that space on your skin and make sure that other bacteria, the ones that could harm you, actually don't come in and invade. So you actually want the ones taking up residence on your skin and so forth, and in your nose and in your eyes, because they are not going to do any harm and they're going to keep the bad guys out. You may have also heard of probiotic therapy. Probiotic therapy is where we use benign and sometimes, if possible, helpful organisms to actually do the same sort of thing. Let's see, I think I've got, Yes. So this is lactobacillus. I have a bunch of stuffed microbes. We would have played with these in class if we were there. So this is lactobacillus acidophilus. You can see he's one of the rod tic-tac bacteria. And this is the one that's most commonly found in yogurt. So this is why it's sometimes considered a probiotic, and why yogurt is often prescribed after taking antibiotics, because it can at least take up space in your gut so that other harmful bacteria won't come in and invade, while your own gut flora are actually reestablishing themselves. So the other problem, the ones that are harmful, we call pathogenic. So pathogens are any sort of organism that cause disease. So what are some examples of these? So I've got all kinds of stuffed organisms here, so I'm going to show you some of them. I've got chlamydia. So this is a pathogenic bacteria. You may have heard of this one. Also got gangrene. So gangrene, chlamydia. Let's see. Here's one that you'd recognize, E. coli. So you've probably heard of E. coli in contaminated fruits and vegetables and meat recently. Let's see, who else do I have? This is H. pylori. This is the bacteria who you can see has got little flagella. Maybe you can. There we go. It's got little hair, little flagella. This is the one that we've linked to ulcers. And let's see, I've got one more. There he is. This is syphilis. He's one of the little spiral bacteria. So there are a good number of pathogenic bacteria, at least ones that you recognize. I don't have the stuffed version of the one that's up here on the map, which is cholera. But the thing is that the number of pathogenic bacteria is small when we compare it to the total number of bacterial species. So there's not actually that many harmful bacteria compared to the benign or helpful bacteria. So real quick, what the diagram over here is showing you, you can see Dr. John Snow. He was one of the first guys to actually figure out pathogens and how they were being transferred. What he mapped out was the incidence, so the bigger the circle, the more deaths there were from cholera. And what he was able to do is build this map and figure out that most of the deaths were very close to this water pump right here. And that showed that this water pump was the source of the cholera infection. And this is what actually is being done today with different diseases. We do these sorts of maps to try and track back where the infections are coming from. In this case, they took the pump off of the handle off the water pump, and cholera incidence went down. If you're not familiar with cholera, one of the main symptoms of cholera is diarrhea. So you actually basically die from dehydration with that sort of disease. Thankfully, today in the United States, we no longer have outbreaks of cholera because most of the time it's due to poor water sanitation. So that's no longer an issue for us here. There are some bacteria which are really interesting. So this little guy right here, you might be able to see he has a knife and fork on him. This is the guy that's here, the Streptococcus pyogenes. This one is normally harmless. So you normally have this one just hanging out in your mouth, in your skin. But this one, some strains of them actually are responsible, as you can see here, for strep throat, scarlet fever, and why that one has a knife and fork, for being a flesh-eating bacteria, necrotizing fasciitis. All right, so I'm going to finish up on this one, and I'll be back to talk a little bit more about bacteria, and then we'll pick up with our next group, the archaeas.